Section 22 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4 by Henry Gray. Composition and Connections of Cranial Nerves, Part 4. The olfactory nerves, first cranial, or nerves of smell, arise from spindle-shaped bipolar cells in the surface epithelium of the olfactory region of the nasal cavity. The non-medullated axons pass upward in groups through numerous foramina in the cribriform plate to the olfactory bulb. Here several fibers, each ending in a tuft of terminal filaments, come into relation with the brush-like ends of a single dendrite from a mitral cell. This interlacing gives rise to the olfactory glomeruli of the bulb. The termination of several or many olfactory fibers in a single glomerulus, where they form synapses with the dendrites of one or two mitral cells, provides for the summation of stimuli in the mitral cells, and accounts in part at least for the detection by the olfactory organs of very dilute solutions. Lateral arborizations of the dendrites of the mitral cells, and the connection of neighboring glomeruli by the axons of small cells of the glomeruli, and the return of impulses of the mitral cells by collaterals, either directly or through the interpellation of granule cells to the dendrites of the mitral cells, reinforce the discharge of the mitral cells along their axons. The axons turn abruptly backward in the deep fiber layer of the bulb to form the olfactory tract. The olfactory tract is continued into the olfactory trigone, just in front of the anterior perforated substance. The axons of the mitral cells on reaching the olfactory trigone separate into three bundles: the lateral olfactory stria, the medial olfactory stria, and the less marked intermedial olfactory stria. The lateral olfactory stria curve lateralward. A few of the fibers end in the olfactory trigone and the anterolateral portion of the anterior perforated substance. Most of the fibers, however, pass into the uncus, the anterior end of the hippocampal gyrus, and there end in the complicated cortex of the hippocampal gyri. The lateral striae more or less disappear as they cross the anterolateral region of the anterior perforated substance. The greater mass of the fibers of the olfactory tract pass into the lateral stria. Numerous collaterals are given into the plexiform layer of the subfrontal cortex, over which the striae pass on their way to the uncus, where they intermingle with the apical dendrons of the medium-sized and small pyramidal cells of the pyramidal layer of this subfrontal or frontal olfactory cortex. The axons give rise to projection fibers, which take an antero-posterior direction to the subthalamic region, sending collaterals and terminal branches to the stria medullaris and others toward the thalamus. Some of the fibers extend farther back and are believed to reach the pons and the medulla oblongata. Most of the fibers of the lateral olfactory stria pass to the hippocampal region of the cortex, especially to the gyrus hippocampi, which may be regarded as the main ending place of the secondary olfactory path derived from axons of the mitral cells. The fibers of the medial olfactory striae terminate for the most part in the parolfactory area, Broca's area. A few end in the subcallosal gyrus, and a few in the anterior perforated substance and the adjoining part of the septum pellucidum. Some of the fibers pass into the anterior commissure, pars olfactoria, to the olfactory tract of the opposite side, where they end partly within the granular layer and partly in the neighborhood of the glomeruli of the olfactory bulb, thus connecting the bulbs of the two sides. The intermediate olfactory striae are as a rule scarcely visible. The fibers terminate in the anterior perforated substance. A few are said to continue to the uncus. The trigonum olfactorium, anterior perforated substance, and the adjoining part of the septum pellucidum are important primary olfactory centres, especially for olfactory reflexes. In these centres terminate many axons from the mitral cells of the olfactory bulb. In addition, the grey substance of the olfactory tract and the gyrus subcallosus receive terminals of the mitral cells. The pathways from these centres to lower centres in the brain stem and spinal cord are only partly known. The most direct path, the tractus olfactor mesencephalicus, basal olfactory bundle of Wallenberg, is supposed to arise from cells in the grey substance of the olfactory tract, the olfactory trigone, the anterior perforated substance, and the adjoining part of the septum pellucidum. The fibres are said to pass direct to the tuber cinereum, to the corpus mammillare, to the brain stem, and the spinal cord. The fibres which enter the mammillary body probably come into relation with cells whose axons give rise to the fasciculus mammilotegmentalis, mammilotegmental bundle of Gooden, which is supposed to end in the grey substance of the tegmentum and of the aqueduct. Some of its fibres are said to join the posterior longitudinal bundle, and others to extend as far as the reticular formation of the pons. Some of the fibres of the medial olfactory stria come into relation with cells in the parolfactory area of Broca and in the anterior perforated substance, whose axons course in the medullary stria of the thalamus. As the axons pass through the lower part of the septum pellucidum, they are joined by other fibres whose cells receive impulses from the mitral cells. These fibres of the medullary stria end for the most part in the habenular nucleus of the same side. Some, however, cross in the habenular commissure, dorsal part of the posterior commissure, to the habenular nucleus of the opposite side. A few fibres of the medullary stria are said to pass by the habenular nucleus to the roof of the midbrain, especially the superior colliculus, while a few others come into relation with the posterior longitudinal bundle and association tracts of the mesencephalon. The ganglion of the habenuli, located in the trigonum habenuli, just in front of the superior colliculus, contain a mesial nucleus with small cells and a lateral nucleus with larger cells. The axons of these cells are grouped together in a bundle, fasciculus retroflexus of Maynard, which passes ventrally medial to the red nucleus and terminates in a small medial ganglion in the substantia perforata posterior, immediately in front of the pons, called the interpeduncular ganglion. The interpeduncular ganglion has rather large nerve cells whose axons curve backward and downward as the tegmental bundle of Gooden, to end partly in the dorsal tegmental nucleus and surrounding grey substance, where they come into relation with association neurons and the dorsal longitudinal bundle of Schutz. The majority of the axons that arise from the mitral cells of the olfactory bulb, and course in the olfactory tract, course in the lateral olfactory stria to the uncus and hippocampal gyrus, and terminate in the cortex. Other fibres probably pass to the uncus and hippocampal gyrus from the primary olfactory centres in the trigonum and anterior perforated substance. The gyrus hippocampus is continued through the isthmus to the gyrus cinguli, which passes over the corpus callosum to the area parolfactoria. The cortical portions of these gyri are connected together by a thick association bundle, the cingulum, that lies buried in the depth of the gyrus cinguli, extending forward to the parolfactory area and backward into the hippocampal region. The axons from the gyrus cinguli pass into the cingulum, many of them bifurcate. The anterior branches together with the axons which run in that direction are traceable as far forward as the anterior part of the septum pellucidum and the anterior end of the corpus striatum, where some of them are incorporated with projection fibres passing toward the internal capsule. The branches and axons which pass backward terminate partly in the hippocampus, the dentate gyrus, and the hippocampal gyrus. Shorter association fibres connect various sections of the gyrus fornicatus, cingulate gyrus, isthmus, and hippocampal gyrus, and these with other regions of the cortex. These gyri constitute the cortical centre for smell. The dentate gyrus, which may be considered as a modified part of the hippocampus, is partially separated from the gyrus hippocampus by the hippocampal fissure and from the fimbria by the fimbrio-dentate sulcus. It is intimately connected with the hippocampal gyrus and the hippocampus. When followed backward, the dentate gyrus separates from the fimbria at the splenium, loses its incisions and knobs, and as the fasciola cinerea passes over the splenium onto the dorsal surface of the corpus callosum and spreads out into a thin layer of grey substance known as the indusium, which can be traced forward around the genu of the corpus callosum into the gyrus subcallosus. The white matter of the indusium, known as the medial longitudinal striae, nerves of Lancisi, and the lateral longitudinal striae are related to the indusium somewhat as the cingulum is to the gyrus cinguli. Axons from the indusium pass into the longitudinal striae, some running forward and others running backward, while some, after entering the medial longitudinal stria, pierce the corpus callosum to join the fornix. Some of the fibres which pass forward extend around the front of the corpus callosum and the anterior commissure, then curve downward, according to Cahal, enter the corpus striatum where they join the olfactory projection path. Other fibres are said to arise in the parolfactory area, the gyrus subcallosus and the anterior perforated substance, diagonal band of Broca, and course backward in the longitudinal striae to the dentate gyrus and the hippocampal region. The indusium is usually considered as a rudimentary part of the rhinencephalon. The olfactory projection fibres which arise from the pyramidal cells of the uncus and hippocampus and from the polymorphic cells of the dentate gyrus form a dense stratum on the ventricular surface, especially on the hippocampus, called the alveus. These fibres pass over into the fimbria and are continued into the fornix. About 1/4 of all the fibres of the fimbria are large projection fibres. The other 3/4 consist of fine commissural fibres which pass from the hippocampus of one side through the fimbria and hippocampal commissure, ventral psalterium or lyra, to the fimbria and hippocampus of the opposite side, where they penetrate the pyramidal layer and terminate in the stratum radiatum. The fibres which course in the fornix pass forward and downward into the corpora mammillare, where numerous collaterals are given off and a few terminate. Most of the fibres in the fornix, however, pass through the corpora, cross the middle line and turn downward in the reticular formation in which they are said to be traceable as far as the pons and possibly farther. As the fornix passes beneath the corpus callosum, it receives fibres from the longitudinal striae of the indusium and from the cingulum. These are the perforating fibres of the fornix which pass through the corpus callosum and course in the fornix toward the mammillary body. As the fornix passes the anterior end of the thalamus, a few fibres are given off to the stria medullaris of the thalamus, and turn back in the stria to the habenular ganglion of the same and the opposite side, having probably the same relation that the reflex fibres have which arise from the primary centres and course in the stria medullaris of the thalamus. Aside from the fibres of the fornix, which pass through the mammillary body to decussate and descend, as the mammulo-mesencephalic fasciculus, many fibres are said to pass into the bundle of Vix-Zazir, and one bundle of fibres is said to pass from the fornix to the tubus cinereum. The mammillary bodies receive collaterals and terminals, then, from the cortical centres via the fornix, and probably other collaterals and terminals are received directly from the primary centres through the tractus olfacto-mesencephalicus. According to Cahal, fibres also reach the mammillary body through the peduncle of the corpus mammillare, from the arcuate fibres of the tegmentum and from the main fillet. The fornix probably brings the cortical centres into relation with the reflex path that runs from the primary centres to the mammillary body and the tubus cinereum. The bundle of Vix-Zazir, mammillothalamic fasciculus, arises from cells in both the medial and lateral nuclei of the mammillary body and by fibres that are directly continued from the fornix. There axons divide within the grey matter. The coarser branches pass into the anterior nucleus of the thalamus as the bundle of Vix-Zazir. The finer branches pass downward as the mammillotegmental bundle of Gudden. The bundle of Vix-Zazir spreads out fan-like as it terminates in the anterior or dorsal nucleus of the thalamus. A few of the fibres pass through the dorsal nucleus to the angular nucleus of the thalamus. The axons from these nuclei are supposed to form part of the thalamocortical system. The mammillotegmental bundle has already been considered under the olfactory reflex paths. The amygdaloid nucleus and the taenia semicircularis, stria terminalis, probably belong to the central olfactory apparatus. The taenia semicircularis extends from the region of the anterior perforated substance to the nucleus amygdalae. Its anterior connections are not clearly understood. Fibres are said to arise from cells in the anterior perforated substance. Some of the fibres pass in front of the anterior commissure, others join the fornix for a short distance as they pass behind the anterior commissure. The two strands ultimately join to form the taenia and pass backward in the groove between the caudate nucleus and the thalamus to the amygdaloid nucleus. Other fibres are said to pass in the opposite direction from the amygdaloid nucleus to the thalamus. End of Part 22.
3 of Gray's Anatomy Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body Part 4 by Henry Gray. Pathways from the brain to the spinal cord. The descending fasciculi which convey impulses from the higher centers to the spinal cord and located in the lateral and ventral funiculi. The motor tract, conveying voluntary impulses, arises from the pyramid cells situated in the motor area of the cortex, the anterior, central, and the posterior portions of the frontal gyri, and the paracentral lobule. The fibers are at first somewhat widely diffused, but as they descend through the corona radiata, they gradually approach each other and pass between the lentiform nucleus and thalamus, in the genu and anterior two-thirds of the occipital part of the internal capsule. Those in the genu are named the geniculate fibers, while the remainder constitute the cerebrospinal fibers. Proceeding downward, they enter the middle 3/5 of the base of the cerebral peduncle. The geniculate fibers cross the middle line and end by arborizing around the cells of the motor nuclei of the cranial nerves. The cerebrospinal fibers are continued downward into the pyramids of the medulla oblongata, and the transit of the fibers from the medulla oblongata is effected by two paths. The fibers nearest to the anterior median fissure cross the middle line, forming the decussation of the pyramids, and descend in the opposite side of the medulla spinalis as the lateral cerebrospinal fasciculus. Cross-pyramidal tract. Throughout the length of the medulla spinalis, fibers from this column pass into the gray substance, to terminate either directly or indirectly around the motor cells of the anterior column. The more laterally placed portion of the tract does not decussate in the medulla oblongata, but descends as the anterior cerebrospinal fasciculus, direct pyramidal tract. These fibers, however, end in the anterior gray column of the opposite side of the medulla spinalis by passing across in the anterior white commissure. There is considerable variation in the extent to which decussation takes place in the medulla oblongata. About 2/3 or 3/4 of the fibers usually decussate in the medulla oblongata and the remainder in the medulla spinalis. The axons of the motor cells in the anterior column pass out as the fibers of the anterior roots of the spinal nerves, along which the impulses are conducted to the muscles of the trunk and limbs. From this it will be seen that all the fibers of the motor tract pass to the nuclei of the motor nerves on the opposite side of the brain or medulla spinalis, a fact which explains why a lesion involving the motor area of one side causes paralysis of the muscles of the opposite side of the body. Further, it will be seen that there is a break in the continuity of the motor chain. In the case of the cranial nerves, this break occurs in the nuclei of these nerves, and in the case of the spinal nerves, in the anterior gray column of the medulla spinalis. For clinical purposes, it is convenient to emphasize this break and divide the motor tract into two portions. One, a series of upper motor neurons, which comprises the motor cells in the cortex and their descending fibers down to the nuclei of the motor nerves. 2, a series of lower motor neurons, which includes the cells of the nuclei of the motor cerebral nerves or the cells of the anterior columns of the medulla spinalis and their axis cylinder processes to the periphery. The rubrospinal fasciculus arises from the large cells of the red nucleus. The fibers cross the raphe of the midbrain in the decussation of Forel and descend in the formatio reticularis of the pons and medulla dorsal to the medial lemniscus, and as they pass into the spinal cord, come to lie in a position ventral to the cross-pyramidal tracts in the lateral funiculus. The rubrospinal fibers end either directly or indirectly by terminals and collaterals about the motor cells in the anterior column on the side opposite from their origin in the red nucleus. A few are said to pass down on the same side. Since the red nucleus is intimately related to the cerebellum by terminals and collaterals of the superior peduncle which arises in the dentate nucleus of the cerebellum, the rubrospinal fasciculus is supposed to be concerned with cerebellar reflexes, complex motor coordinations necessary in locomotion and equilibrium. The afferent paths concerned in these reflexes have already been partly considered, namely, the dorsal and ventral spinocerebellar fasciculi, and probably some of the fibers of the posterior funiculi which reach the cerebellum by the inferior peduncle. The tectospinal fasciculus arises from the superior colliculus of the roof tectum of the midbrain. The axons come from large cells in the stratum opticum and stratum lemnici and sweep ventrally around the central gray matter of the aqueduct, cross the raphe in the fountain decussation of Maynard, and turn downward in the tegmentum in the ventral longitudinal bundle. Some of the fibers do not cross in the raphe, but pass down on the same side. It is uncertain whether they come from the superior colliculus of the same side or arch over the aqueduct from the colliculus of the opposite side. The tectospinal fasciculus, which comprises the major part of the ventral longitudinal bundle, passes down through the tegmentum and reticular formation of the pons and medulla oblongata ventral to the medial longitudinal bundle. In the medulla, the two bundles are more or less intermingled, and the tectospinal portion is continued into the anterolateral funiculus of the spinal cord ventral to the rubrospinal fasciculus, with which some of its fibers are intermingled. Some of the fibers of the tectospinal fasciculus pass through the red nucleus, giving off collaterals to it. Others are given off to the motor nuclei of the cranial nerves, and in the spinal cord they terminate either directly or indirectly by terminals and collaterals among the nuclei of the anterior column. Since the superior colliculus is an important optic reflex center, this tract is probably concerned in optic reflexes, and possibly also with auditory reflexes, since some of the fibers of the central auditory path, the lateral lemniscus, terminate in the superior colliculus. The vestibulospinal fasciculus, part of the anterior marginal fasciculus or Lowenthal's tract, situated chiefly in the marginal part of the anterior funiculus, is mainly derived from the cells of the terminal nuclei of the vestibular nerve, probably Dieter's and Bechtereau's, and some of its fibers are supposed to come from the nucleus fastigius, roof nucleus of the cerebellum. The latter nucleus is intimately connected with Dieter's and Bechtereau's nuclei. The vestibulospinal fasciculus is concerned with equilibratory reflexes. Its terminals and collaterals end about the motor cells in the anterior column. It extends to the sacral region of the cord. Its fibers are intermingled with the ascending spinothalamic fasciculus, with the anterior proper fasciculus, and laterally with the tectospinal fasciculus. Its fibers are supposed to be both crossed and uncrossed. In the brain stem it is associated with the dorsal longitudinal bundle. The pontospinal fasciculus, Bechtereau, arises from the cells in the reticular formation of the pons from the same and the opposite side, and is associated in the brain stem with the ventral longitudinal bundle. In the cord it is intermingled with the fibers of the vestibulospinal fasciculus in the anterior funiculus. Not much is known about this tract. There are probably other descending fasciculi, such as the thalamospinal, but not much is known about them. End of section 23.
Of Grey's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Read by Laurie Anne Walden. Anatomy of the Human Body, Part 4, by Henry Gray. The Meninges of the Brain and Medulla Spinalis, Part 1. The brain and medulla spinalis are enclosed within three membranes. These are named from without inward, the dura mater, the arachnoid, and the pia mater. The dura mater. The dura mater is a thick and dense inelastic membrane. The portion which encloses the brain differs in several essential particulars from that which surrounds the medulla spinalis, and therefore it is necessary to describe them separately. But at the same time it must be distinctly understood that the two form one complete membrane, and are continuous with each other at the foramen magnum. The cranial dura mater encephali, dura of the brain, lines the interior of the skull, and serves the twofold purpose of an internal periosteum to the bones, and a membrane for the protection of the brain. It is composed of two layers, an inner or meningeal, and an outer or endosteal, closely connected together, except in certain situations, where, as already described, they separate to form sinuses for the passage of venous blood. Its outer surface is rough and fibrillated and adheres closely to the inner surfaces of the bones, the adhesions being most marked opposite the sutures and at the base of the skull. Its inner surface is smooth and lined by a layer of endothelium. It sends inward 4 processes which divide the cavity of the skull into a series of freely communicating compartments for the lodgment and protection of the different parts of the brain, and it is prolonged to the outer surface of the skull through the various foramina which exist at the base, and thus becomes continuous with the pericranium. Its fibrous layer forms sheaths for the nerves which pass through these apertures. Around the margin of the foramen magnum it is closely adherent to the bone, and is continuous with the spinal dura mater. Processes. The processes of the cranial dura mater, which projects into the cavity of the skull, are formed by reduplications of the inner or meningeal layer of the membrane, and are 4 in number: the falx cerebri, the tentorium cerebelli, the falx cerebelli, and the diaphragma celli. The falx cerebri, so named from its sickle-like form, is a strong, arched process which descends vertically in the longitudinal fissure between the cerebral hemispheres. It is narrow in front, where it is attached to the crista galli of the ethmoid, and broad behind, where it is connected with the upper surface of the tentorium cerebelli. Its upper margin is convex and attached to the inner surface of the skull in the middle line, as far back as the internal occipital protuberance. It contains the superior sagittal sinus. Its lower margin is free and concave, and contains the inferior sagittal sinus. The tentorium cerebelli is an arched lamina, elevated in the middle and inclining downward toward the circumference. It covers the superior surface of the cerebellum and supports the occipital lobes of the brain. Its anterior border is free and concave, and bounds a large oval opening, the incisura tentorii, for the transmission of the cerebral peduncles. It is attached, behind, by its convex border, to the transverse ridges upon the inner surface of the occipital bone, and there encloses the transverse sinuses; in front, to the superior angle of the petrous part of the temporal bone on either side, enclosing the superior petrosal sinuses. At the apex of the petrous part of the temporal bone the free and attached borders meet, and crossing one another are continued forward to be fixed to the anterior and posterior clinoid processes respectively. To the middle line of its upper surface the posterior border of the falx cerebri is attached, the straight sinus being placed at their line of junction. The falx cerebelli is a small triangular process of dura mater, received into the posterior cerebellar notch. Its base is attached, above, to the under and back part of the tentorium, its posterior margin to the lower division of the vertical crest on the inner surface of the occipital bone. As it descends it sometimes divides into 2 smaller folds, which are lost on the sides of the foramen magnum. The diaphragma celli is a small circular horizontal fold which roofs in the sella turcica and almost completely covers the hypophysis. A small central opening transmits the infundibulum. Structure. The cranial dura mater consists of white fibrous tissue and elastic fibers arranged in flattened laminae which are imperfectly separated by lacunar spaces and blood vessels into 2 layers, endosteal and meningeal. The endosteal layer is the internal periosteum for the cranial bones, and contains the blood vessels for their supply. At the margin of the foramen magnum it is continuous with the periosteum lining the vertebral canal. The meningeal or supporting layer is lined on its inner surface by a layer of nucleated flattened mesothelium, similar to that found on serous membranes. The arteries of the dura mater are very numerous. Those in the anterior fossa are the anterior meningeal branches of the anterior and posterior ethmoidal and internal carotid, and a branch from the middle meningeal. Those in the middle fossa are the middle and accessory meningeal of the internal maxillary, a branch from the ascending pharyngeal, which enters the skull through the foramen lacerum, branches from the internal carotid, and a recurrent branch from the lacrymal. Those in the posterior fossa are meningeal branches from the occipital, one entering the skull through the jugular foramen, and another through the mastoid foramen, the posterior meningeal from the vertebral, occasional meningeal branches from the ascending pharyngeal, entering the skull through the jugular foramen and hypoglossal canal, and a branch from the middle meningeal. The veins returning the blood from the cranial dura mater anastomose with the diploic veins and end in the various sinuses. Many of the meningeal veins do not open directly into the sinuses, but indirectly through a series of ampullae, termed venous lacunae. These are found on either side of the superior sagittal sinus, especially near its middle portion, and are often invaginated by arachnoid granulations. They also exist near the transverse and straight sinuses. They communicate with the underlying cerebral veins, and also with the diploic and emissary veins. The nerves of the cranial dura mater are filaments from the semilunar ganglion, from the ophthalmic, maxillary, mandibular, vagus, and hypoglossal nerves, and from the sympathetic. The spinal dura mater spinalis, spinal dura, forms a loose sheath around the medulla spinalis, and represents only the inner or meningeal layer of the cranial dura mater. The outer or endosteal layer ceases at the foramen magnum, its place being taken by the periosteum lining the vertebral canal. The spinal dura mater is separated from the arachnoid by a potential cavity, the subdural cavity. The two membranes are, in fact, in contact with each other, except where they are separated by a minute quantity of fluid which serves to moisten the opposed surfaces. It is separated from the wall of the vertebral canal by a space, the epidural space, which contains a quantity of loose areolar tissue and a plexus of veins. The situation of these veins between the dura mater and the periosteum of the vertebrae corresponds, therefore, to that of the cranial sinuses between the meningeal and endosteal layers of the cranial dura mater. The spinal dura mater is attached to the circumference of the foramen magnum and to the second and third cervical vertebrae. It is also connected to the posterior longitudinal ligament, especially near the lower end of the vertebral canal, by fibrous slips. The subdural cavity ends at the lower border of the second sacral vertebra. Below this level the dura mater closely invests the filum terminale and descends to the back of the coccyx, where it blends with the periosteum. The sheath of dura mater is much larger than is necessary for the accommodation of its contents, and its size is greater in the cervical and lumbar regions than in the thoracic. On each side may be seen the double openings which transmit the two roots of the corresponding spinal nerve, the dura mater being continued in the form of tubular prolongations on them as they pass through the intervertebral foramina. These prolongations are short in the upper part of the vertebral column, but gradually become longer below, forming a number of tubes of fibrous membrane which enclose the lower spinal nerves and are contained in the vertebral canal. Structure. The spinal dura mater resembles in structure the meningeal or supporting layer of the cranial dura mater, consisting of white fibrous and elastic tissue arranged in bands or lamellae, which, for the most part, are parallel with one another and have a longitudinal arrangement. Its internal surface is smooth and covered by a layer of mesothelium. It is sparingly supplied with blood vessels, and a few nerves have been traced into it. The arachnoid. The arachnoid is a delicate membrane enveloping the brain and medulla spinalis, and lying between the pia mater internally and the dura mater externally. It is separated from the pia mater by the subarachnoid cavity, which is filled with cerebrospinal fluid. The cranial part, arachnoidea encephali, of the arachnoid invests the brain loosely, and does not dip into the sulci between the gyri, nor into the fissures, with the exception of the longitudinal. On the upper surface of the brain the arachnoid is thin and transparent; at the base it is thicker, and slightly opaque toward the central part, where it extends across between the two temporal lobes in front of the pons, so as to leave a considerable interval between it and the brain. The spinal part, arachnoidea spinalis, of the arachnoid is a thin, delicate, tubular membrane loosely investing the medulla spinalis. Above it is continuous with the cranial arachnoid. Below it widens out and invests the cauda equina and the nerves proceeding from it. It is separated from the dura mater by the subdural space, but here and there this space is traversed by isolated connective tissue trabeculae, which are most numerous on the posterior surface of the medulla spinalis. The arachnoid surrounds the cranial and spinal nerves, and encloses them in loose sheaths as far as their points of exit from the skull and vertebral canal. Structure. The arachnoid consists of bundles of white fibrous and elastic tissue intimately blended together. Its outer surface is covered with a layer of low cuboidal mesothelium. The inner surface and the trabeculae are likewise covered by a somewhat low type of cuboidal mesothelium, which in places are flattened to a pavement type. Vessels of considerable size, but few in number, and, according to Bachtelek, a rich plexus of nerves derived from the motor root of the trigeminal, the facial, and the accessory nerves, are found in the arachnoid. The subarachnoid cavity, cavum subarachnoideale, subarachnoid space, is the interval between the arachnoid and pia mater. It is occupied by a spongy tissue consisting of trabeculae of delicate connective tissue and intercommunicating channels in which the subarachnoid fluid is contained. This cavity is small on the surface of the hemispheres of the brain. On the summit of each gyrus the pia mater and the arachnoid are in close contact, but in the sulci between the gyri triangular spaces are left, in which the subarachnoid trabecular tissue is found, for the pia mater dips into the sulci, whereas the arachnoid bridges across them from gyrus to gyrus. At certain parts of the base of the brain the arachnoid is separated from the pia mater by wide intervals which communicate freely with each other and are named subarachnoid cisternae. In these the subarachnoid tissue is less abundant. End of section 24. of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Read by Laurie Anne Walden. Anatomy of the Human Body, Part 4 by Henry Gray. The Meninges of the Brain and Medulla Spinalis, Part 2. Subarachnoid cisternae, cisternae subarachnoidales. The cisterna cerebellomedullaris, cisterna magna, is triangular on sagittal section, and results from the arachnoid bridging over the interval between the medulla oblongata and the under surfaces of the hemispheres of the cerebellum. It is continuous with the subarachnoid cavity of the medulla spinalis at the level of the foramen magnum. The cisterna pontis is a considerable space on the ventral aspect of the pons. It contains the basilar artery, and is continuous behind with the subarachnoid cavity of the medulla spinalis, and with the cisterna cerebellomedullaris, and in front of the pons with the cisterna interpeduncularis. The cisterna interpeduncularis, cisterna basalis, is a wide cavity where the arachnoid extends across between the two temporal lobes. It encloses the cerebral peduncles and the structures contained in the interpeduncular fossa, and contains the arterial circle of Willis. In front, the cisterna interpeduncularis extends forward across the optic chiasma forming the cisterna chiasmatis, and onto the upper surface of the corpus callosum, for the arachnoid stretches across from one cerebral hemisphere to the other immediately beneath the free border of the falx cerebri, and thus leaves a space in which the anterior cerebral arteries are contained. The cisterna fossae cerebri lateralis is formed in front of either temporal lobe by the arachnoid bridging across the lateral fissure. This cavity contains the middle cerebral artery. The cisterna veni magni cerebri occupies the interval between the splenium of the corpus callosum and the superior surface of the cerebellum. It extends between the layers of the tela choroidea of the third ventricle, and contains the great cerebral vein. The subarachnoid cavity communicates with the general ventricular cavity of the brain by three openings. One, the foramen of Magendie, is in the middle line at the inferior part of the roof of the fourth ventricle. The other two are at the extremities of the lateral recesses of that ventricle, behind the upper roots of the glossopharyngeal nerves, and are known as the foramina of Luschka. It is still somewhat uncertain whether these foramina are actual openings or merely modified areas of the inferior velum which permit the passage of the cerebrospinal fluid from the ventricle into the subarachnoid spaces as through a permeable membrane. The spinal part of the subarachnoid cavity is a very wide interval, and is the largest at the lower part of the vertebral canal, where the arachnoid encloses the nerves which form the cauda equina. Above it is continuous with the cranial subarachnoid cavity; below it ends at the level of the lower border of the second sacral vertebra. It is partially divided by a longitudinal septum, the subarachnoid septum, which connects the arachnoid with the pia mater opposite the posterior median sulcus of the medulla spinalis, and forms a partition, incomplete and cribriform above, but more perfect in the thoracic region. The spinal subarachnoid cavity is further subdivided by the ligamentum denticulatum, which will be described with the pia mater. The cerebrospinal fluid is a clear, limpid fluid, having a saltish taste, and a slightly alkaline reaction. According to Lassagne, it consists of 98.5 parts of water, the remaining 1.5% being solid matters, animal and saline. It varies in quantity, being most abundant in old persons, and is quickly secreted. The arachnoidvilli, granulationes arachnoidiales, glandulae pacchioni, pacchionian bodies, are small, fleshy-looking elevations, usually collected into clusters of variable size, which are present upon the outer surface of the dura mater, in the vicinity of the superior sagittal sinus, and in some other situations. Upon laying open the sagittal sinus and the venous lacunae on either side of it, villi will be found protruding into its interior. They are not seen in infancy, and very rarely until the 3rd year. They are usually found after the 7th year, and from this period they increase in number and size as age advances. They are not glandular in structure, but are enlarged normal villi of the arachnoid. As they grow they push the thinned dura mater before them, and cause absorption of the bone from pressure, and so produce the pits or depressions on the inner wall of the calvarium. Structure. An arachnoidal villus represents an invasion of the dura by the arachnoid membrane. The latter penetrates the dura in such a manner that the arachnoid mesothelial cells come to lie directly beneath the vascular endothelium of the great dural sinuses. It consists of the following parts. 1. In the interior is a core of subarachnoid tissue, continuous with the meshwork of the general subarachnoid tissue through a narrow pedicle by which the villus is attached to the arachnoid. 2. Around this tissue is a layer of arachnoid membrane, limiting and enclosing the subarachnoid tissue. 3. Outside this is the thinned wall of the lacuna, which is separated from the arachnoid by a potential space which corresponds to and is continuous with the subdural cavity. 4. And finally, if the villus projects into the sagittal sinus, it will be covered by the greatly thinned wall of the sinus, which may consist merely of endothelium. It will be seen, therefore, that fluid injected into the subarachnoid cavity will find its way into these villi, and it has been found experimentally that it passes from the villi into the venous sinuses, into which they project. The pia mater. The pia mater is a vascular membrane consisting of a minute plexus of blood vessels, held together by an extremely fine areolar tissue and covered by a reflection of the mesothelial cells from the arachnoid trabeculae. It is an incomplete membrane, absent probably at the foramen of Magendie and the 2 foramina of Luschka, and perforated in a peculiar manner by all the blood vessels as they enter or leave the nervous system. In the perivascular spaces the pia apparently enters as a mesothelial lining of the outer surface of the space. A variable distance from the exterior these cells become unrecognizable and are apparently lacking, replaced by neuroglia elements. The inner walls of these perivascular spaces seem likewise covered, for a certain distance, by the mesothelial cells, reflected with the vessels from the arachnoid covering of these vascular channels as they traverse the subarachnoid spaces. The cranial pia mater encephali, pia of the brain, invests the entire surface of the brain, dips between the cerebral gyri and cerebellar laminae, and is invaginated to form the tela choroidea of the third ventricle, and the choroid plexuses of the lateral and third ventricles. As it passes over the roof of the fourth ventricle, it forms the tela choroidea and the choroid plexuses of this ventricle. On the cerebellum the membrane is more delicate. The vessels from its deep surface are shorter, and its relations to the cortex are not so intimate. The spinal pia mater spinalis, pia of the cord, is thicker, firmer, and less vascular than the cranial pia mater. This is due to the fact that it consists of two layers, the outer, or additional one, being composed of bundles of connective tissue fibers, arranged, for the most part, longitudinally. Between the layers are cleft-like spaces which communicate with the subarachnoid cavity, and a number of blood vessels which are enclosed in perivascular lymphatic sheaths. The spinal pia mater covers the entire surface of the medulla spinalis, and is very intimately adherent to it. In front, it sends a process backward into the anterior fissure. A longitudinal fibrous band, called the linea splendens, extends along the middle line of the anterior surface, and a somewhat similar band, the ligamentum denticulatum, is situated on either side. Between the conus medullaris, the pia mater is continued as a long, slender filament, filum terminale, which descends through the center of the mass of nerves forming the cauda equina. It blends with the dura mater at the level of the lower border of the second sacral vertebra, and extends downward as far as the base of the coccyx, where it fuses with the periosteum. It assists in maintaining the medulla spinalis in its position during the movements of the trunk, and is, from this circumstance, called the central ligament of the medulla spinalis. The pia mater forms sheaths for the cranial and spinal nerves. These sheaths are closely connected with the nerves, and blend with their common membranous investments. The ligamentum denticulatum, dentate ligament, is a narrow fibrous band situated on either side of the medulla spinalis throughout its entire length, and separating the anterior from the posterior nerve roots. Its medial border is continuous with the pia mater at the side of the medulla spinalis. Its lateral border presents a series of triangular, tooth-like processes, the points of which are fixed at intervals to the dura mater. These processes are 21 in number, on either side, the first being attached to the dura mater, opposite the margin of the foramen magnum, between the vertebral artery and the hypoglossal nerve, and the last near the lower end of the medulla spinalis. End of the meninges of the brain and medulla spinalis. The cerebrospinal fluid. The cerebrospinal fluid, for the most part elaborated by the choroid plexuses, is poured into the cerebral ventricles which are lined by smooth ependyma. That portion of the fluid formed in the lateral ventricles escapes by the foramen of Monro into the third ventricle, and thence by the aqueduct into the fourth ventricle. Likewise an ascending current of fluid apparently occurs in the central canal of the spinal cord. This, representing a possible product of the ependyma, may be added to the intraventricular supply. From the fourth ventricle the fluid is poured into the subarachnoid spaces through the medial foramen of Magendie and the two lateral foramina of Luschka. There is no evidence that functional communications between the cerebral ventricles and the subarachnoid spaces exist in any region except from the fourth ventricle. In addition to the elaboration of the cerebrospinal fluid by the choroid plexuses, there seems fairly well established a second source of the fluid from the nervous system itself. The blood vessels that enter and leave the brain are surrounded by perivascular channels. It seems most likely that the outer wall of these channels is lined by a continuation inward of the pial mesothelium, while the inner wall is probably derived from the mesothelial covering of the vessels, which are thus protected throughout the subarachnoid spaces. These mesothelial cells continue inward only a short distance, neuroglia cells probably replacing, on the outer surface, the mesothelial elements. Through these perivascular channels there is probably a small amount of fluid flowing from nerve cell to subarachnoid space. The chemical differences between the subarachnoid fluid, product of choroid plexuses and perivascular system, and the ventricular fluid, product of choroid plexuses alone, indicate that the products of nerve metabolism are poured into the subarachnoid space. The absorption of the cerebrospinal fluid is a dual process, being chiefly a rapid drainage through the arachnoidvilli into the great dural sinuses, and, in small part, a slow escape into the true lymphatic vessels, by way of an abundant but indirect perineural course. In general, the arachnoid channels are equipped as fluid retainers with unquestionable powers of diffusion or absorption in regard to certain elements in the normal cerebrospinal fluid, deriving in this way a cellular nutrition. The subdural space, between arachnoid and dura, is usually considered to be a part of the cerebrospinal channels. It is a very small space, the two limiting surfaces being separated by merely a capillary layer of fluid. Whether this fluid is exactly similar to the cerebrospinal fluid is very difficult to ascertain. Likewise, our knowledge of the connections between the subdural and subarachnoid spaces is hardly definite. In some ways, the subdural space may be likened to a serous cavity. The inner surface of the dura is covered by flattened polygonal mesothelial cells, but the outer surface of the arachnoid is covered by somewhat cuboidal mesothelium. The fluid of the subdural space has probably a local origin from the cells lining it. End of section 25.
Section number 26 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4, by Henry Gray. The cranial nerves, nervi cerebralis, cerebral nerves. There are 12 pairs of cranial nerves. They are attached to the brain and are transmitted through foramina in the base of the cranium. The different pairs are named from before backwards as follows: first, olfactory; second, optic; third, oculomotor; fourth, trochlear; fifth, trigeminal; sixth, abducent; seventh, facial; eighth, acoustic; ninth, glossopharyngeal; 10th, vagus; 11th, accessory; 12th, hypoglossal. The area of attachment of a cranial nerve to the surface of the brain is termed its superficial or apparent origin. The fibers of the nerve can be traced into the substance of the brain to a special nucleus of grey substance. The motor or efferent cranial nerves arise within the brain from groups of nerve cells which constitute their nuclei of origin. The sensory or afferent cranial nerves arise from groups of nerve cells outside the brain. These nerve cells may be grouped to form ganglia on the trunks of the nerves, or may be situated in peripheral sensory organs such as the nose and eye. The central processes of these cells run into the brain, and there end by arborizing around nerve cells, which are grouped to form nuclei of termination. The nuclei of origin of the motor nerves and the nuclei of termination of the sensory nerves are brought into relationship with the cerebral cortex, the former through the geniculate fibers of the internal capsule, the latter through the lemniscus. The geniculate fibers arise from the cells of the motor area of the cortex, and, after crossing the middle line, end by arborizing around the cells of the nuclei of origin of the motor cranial nerves. On the other hand, fibers arise from the cells of the nuclei of termination of the sensory nerves, and, after crossing to the opposite side, join the lemniscus, and thus connect these nuclei, directly or indirectly, with the cerebral cortex. 5a. The olfactory nerves. The olfactory nerves, or nerves of smell, are distributed to the mucous membrane of the olfactory region of the nasal cavity. This region comprises the superior nasal concha, and the corresponding part of the nasal septum. The nerves originate from the central or deep processes of the olfactory cells of the nasal mucous membrane. They form a plexiform network in the mucous membrane, and are then collected into about 20 branches, which pierce the cribriform plate of the ethmoid bone in two groups, a lateral and a medial group, and end in the glomeruli of the olfactory bulb. Each branch receives tubular sheaths from the dura mater and pia mater, the former being lost in the periosteum of the nose, the latter in the neuralemma of the nerve. The olfactory nerves are non-medullated, and consist of axis cylinders surrounded by nucleated sheaths, in which, however, there are fewer nuclei than are found in the sheaths of ordinary non-medullated nerve fibres. The olfactory centre in the cortex is generally associated with the rhinencephalon. The olfactory nerves are developed from the cells of the ectoderm which lines the olfactory pits. These cells undergo proliferation and give rise to what are termed the olfactory cells of the nose. The axons of the olfactory cells grow into the overlying olfactory bulb and form the olfactory nerves. 5b. The optic nerve. Nervus opticus. Second nerve. The optic nerve, or nerve of sight, consists mainly of fibres derived from the ganglionic cells of the retina. These axons terminate in arborizations around the cells in the lateral geniculate body, pulvinar, and superior colliculus which constitute the lower or primary visual centres. From the cells of the lateral geniculate body and the pulvinar, fibres pass to the cortical visual centre, situated in the cuneus and in the neighbourhood of the calcarine fissure. A few fibres of the optic nerve, of small calibre, pass from the primary centres to the retina and are supposed to govern chemical changes in the retina, and also the movements of some of its elements, pigment cells and cones. There are also a few fine fibres, afferent fibres, extending from the retina to the brain, that are supposed to be concerned in pupillary reflexes. The optic nerve is peculiar in that its fibres and ganglion cells are probably third in the series of neurons from the receptors to the brain. Consequently, the optic nerve corresponds rather to a tract of fibres within the brain than to the other cranial nerves. Its fibres pass backward and medialward through the orbit and optic foramen to the optic commissure, where they partially decussate. The mixed fibres from the two nerves are continued in the optic tracts, the primary visual centres of the brain. The orbital portion of the optic nerve is from 20 mm to 30 mm in length, and has a slightly sinuous course to allow for movements of the eyeball. It is invested by an outer sheath of dura mater and an inner sheath from the arachnoid which are attached to the sclera around the area where the nerve fibres pierce the choroid and sclera of the bulb. A little behind the bulb of the eye, the central artery of the retina with its accompanying vein perforates the optic nerve, and runs within it to the retina. As the nerve enters the optic foramen, its dural sheath becomes continuous with that lining the orbit and the optic foramen. In the optic foramen, the ophthalmic artery lies below and to its outer side. The intercranial portion of the optic nerve is about 10 mm in length. The optic chiasma, chiasma opticum, somewhat quadrilateral in form, rests upon the tuberculum sellae and on the anterior part of the diaphragma sellae. It is in relation, above, with the lamina terminalis, behind with the tuber cinereum, on either side with the anterior perforated substance. Within the chiasma the optic nerves undergo a partial decussation. The fibres forming the medial part of each tract and posterior part of the chiasma have no connection with the optic nerves. They simply cross in the chiasma, and connect the medial geniculate bodies of the two sides. They form the commissure of Gudden. The remaining and principal part of the chiasma consists of two sets of fibres, crossed and uncrossed. The crossed fibres, which are the more numerous, occupy the central part of the chiasma, and pass from the optic nerve of one side to the optic tract of the other, decussating in the chiasma with similar fibres of the opposite optic nerve. The uncrossed fibres occupy the lateral part of the chiasma, and pass from the nerve of one side into the tract of the same side. Note 130. A specimen of congenital absence of the optic chiasma is to be found in the Museum of the Westminster Hospital. See also Henle, Nervenlehre, page 393, edition 2. The crossed fibres of the optic nerve tend to occupy the medial side of the nerve and the uncrossed fibres the lateral side. In the optic tract, however, the fibres are much more intermingled. The optic tract passes backward and outward from the optic chiasma over the tuber cinereum and anterior perforated space to the cerebral peduncle and winds obliquely across its under surface. Its fibres terminate in the lateral geniculate body, the pulvinar and the superior colliculus. It is adherent to the tuber cinereum and the cerebral peduncle as it passes over them. In the region of the lateral geniculate body it splits into two bands. The medial and smaller one is a part of the commissure of Gudden and ends in the medial geniculate body. From its mode of development and from its structure, the optic nerve must be regarded as a prolongation of the brain substance, rather than as an ordinary cerebrospinal nerve. As it passes from the brain, it receives sheaths from the three cerebral membranes, a perineural sheath from the pia mater, an intermediate sheath from the arachnoid, and an outer sheath from the dura mater, which is also connected with the periosteum as it passes through the optic foramen. These sheaths are separated from each other by cavities which communicate with the subdural and subarachnoid cavities respectively. The innermost or perineural sheath sends a process around the arteria centralis retinae into the interior of the nerve, and enters intimately into its structure. End of section number 26. Of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4, by Henry Gray. 5c. The oculomotor nerve. Nervus oculomotorius, third nerve. The oculomotor nerve supplies somatic motor fibres to all the ocular muscles, except the obliquus superior and rectus lateralis. It also supplies, through its connections with the ciliary ganglion, sympathetic motor fibres to the sphincter pupillae and the ciliaris muscles. The fibres of the oculomotor nerve arise from a nucleus which lies in the grey substance of the floor of the cerebral aqueduct, and extends in front of the aqueduct for a short distance into the floor of the third ventricle. From this nucleus fibres pass through the tegmentum, the red nucleus, and the medial part of the substantia nigra, forming a series of curves with a lateral convexity, and emerge from the oculomotor sulcus on the medial side of the cerebral peduncle. The nucleus of the oculomotor nerve does not consist of a continuous column of cells, but is broken up into a number of smaller nuclei, which are arranged in two groups, anterior and posterior. Those of the posterior group are 6 in number, 5 of which are symmetrical on the two sides of the middle line, while the 6th is centrally placed and is common to the nerves of both sides. The anterior group consists of 2 nuclei, an anteromedial and anterolateral. The nucleus of the oculomotor nerve, considered from a physiological standpoint, can be subdivided into several smaller groups of cells, each group controlling a particular muscle. On emerging from the brain, the nerve is invested with a sheath of pia mater, and enclosed in a prolongation from the arachnoid. It passes between the superior cerebellar and posterior cerebral arteries, and then pierces the dura mater in front of and lateral to the posterior clinoid process, passing between the free and detached borders of the tentorium cerebelli. It runs along the lateral wall of the cavernous sinus, above the other orbital nerves, receiving in its course 1 or 2 filaments from the cavernous plexus of the sympathetic, and a communicating branch from the ophthalmic division of the trigeminal. It then divides into 2 branches, which enter the orbit through the superior orbital fissure, between the 2 heads of the rectus lateralis. Here the nerve is placed below the trochlear nerve and the frontal and lacrimal branches of the ophthalmic nerve, while the nasociliary nerve is placed between its 2 rami. The superior ramus, the smaller, passes medialward over the optic nerve and supplies the rectus superior and levator palpebrae superioris. The inferior ramus, the larger, divides into 3 branches. One passes beneath the optic nerve to the rectus medialis, another to the rectus inferior. The third and longest runs forward between the recti inferior and lateralis to the obliquus inferior. From the last a short, thick branch is given off to the lower part of the ciliary ganglion, and forms its short root. All these branches enter the muscles on their ocular surfaces, with the exception of the nerve to the obliquus inferior, which enters the muscle at its posterior border. 5d. The trochlear nerve. Nervus trochlearis, fourth nerve. The trochlear nerve, the smallest of the cranial nerves, supplies the obliquus superior oculi. It arises from a nucleus situated in the floor of the cerebral aqueduct, opposite the upper part of the inferior colliculus. From its origin it runs downward through the tegmentum, and then turns backward into the upper part of the anterior medullary velum. Here it decussates with its fellow of the opposite side, and emerges from the surface of the velum at the side of the frenulum velli, immediately behind the inferior colliculus. The nerve is directed across the superior cerebellar peduncle, and then winds forward around the cerebral peduncle, immediately above the pons, pierces the dura mater in the free border of the tentorium cerebelli, just behind and lateral to the posterior clinoid process, and passes forward in the lateral wall of the cavernous sinus, between the oculomotor nerve and the ophthalmic division of the trigeminal. It crosses the oculomotor nerve, and enters the orbit through the superior orbital fissure. It now becomes the highest of all the nerves, and lies medial to the frontal nerve. In the orbit it passes medialward, above the origin of the levator palpebrae superioris, and finally enters the orbital surface of the obliquus superior. In the lateral wall of the cavernous sinus the trochlear nerve forms communications with the ophthalmic division of the trigeminal with the cavernous plexus of the sympathetic. In the superior orbital fissure it occasionally gives off a branch to the lacrimal nerve. It gives off a recurrent branch which passes backward between the layers of the tentorium cerebelli and divides into 2 or 3 filaments, which may be traced as far as the wall of the transverse sinus. End of section 27. Of Grey's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4, by Henry Gray. 5e. The trigeminal nerve. Nervus trigeminus, 5th or trifacial nerve. The trigeminal nerve is the largest cranial nerve and is the great sensory nerve of the head and face, and the motor nerve of the muscles of mastication. It emerges from the side of the pons, near its upper border, by a small motor and a large sensory root, the former being situated in front of and medial to the latter. Motor root. The fibres of the motor root arise from 2 nuclei, a superior and an inferior. The superior nucleus consists of a strand of cells occupying the whole length of the lateral portion of the grey substance of the cerebral aqueduct. The inferior or chief nucleus is situated in the upper part of the pons, close to its dorsal surface, and along the line of the lateral margin of the rhomboid fossa. The fibres from the superior nucleus constitute the mesencephalic root. They descend through the midbrain and, entering the pons, join with the fibres from the lower nucleus, and the motor root, thus formed, passes forward through the pons to its point of emergence. It is uncertain whether the mesencephalic root is motor or sensory. Sensory root. The fibres of the sensory root arise from the cells of the semilunar ganglion, which lies in a cavity of the dura mater near the apex of the petrous part of the temporal bone. They pass backward below the superior petrosal sinus and tentorium cerebelli, and, entering the pons, divide into upper and lower roots. The upper root ends partly in a nucleus which is situated in the pons lateral to the lower motor nucleus, and partly in the locus coeruleus. The lower root descends through the pons and medulla oblongata, and ends in the upper part of the substantia gelatinosa of Rolando. This lower root is sometimes named the spinal root of the nerve. Medullation of the fibres of the sensory root begins about the 5th month of fetal life, but the whole of its fibres are not medullated until the 3rd month after birth. The semilunar ganglion. Ganglion semilunare, Gasseri, Gasserian ganglion, occupies a cavity, cavum Merkellii, in the dura mater covering the trigeminal impression near the apex of the petrous part of the temporal bone. It is somewhat crescentic in shape, with its convexity directed forward. Medially it is in relation with the internal carotid artery and the posterior part of the cavernous sinus. The motor root runs in front and medial to the sensory root, and passes beneath the ganglion. It leaves the skull through the foramen ovale, and, immediately below this foramen, joins the mandibular nerve. The greater superficial petrosal nerve lies also underneath the ganglion. The ganglion receives, on its medial side, filaments from the carotid plexus of the sympathetic. It gives off minute branches to the tentorium cerebelli, and to the dura mater in the middle fossa of the cranium. From its convex border, which is directed forward and lateralward, 3 large nerves proceed, viz., the ophthalmic, maxillary, and mandibular. The ophthalmic and maxillary consist exclusively of sensory fibres. The mandibular is joined outside the cranium by the motor root. Associated with the 3 divisions of the trigeminal nerve are 4 small ganglia. The ciliary ganglion is connected with the ophthalmic nerve, the sphenopalatine ganglion with the maxillary nerve, and the otic and submaxillary ganglia with the mandibular nerve. All 4 receive sensory filaments from the trigeminal, and motor and sympathetic filaments from various sources. These filaments are called the roots of the ganglia. The ophthalmic nerve, nervus ophthalmicus, or first division of the trigeminal, is a sensory nerve. It supplies branches to the cornea, ciliary body and iris, to the lacrimal gland and conjunctiva, to the part of the mucous membrane of the nasal cavity, and to the skin of the eyelids, eyebrow, forehead, and nose. It is the smallest of the 3 divisions of the trigeminal, and arises from the upper part of the semilunar ganglion as a short, flattened band about 2.5 cm long, which passes forward along the lateral wall of the cavernous sinus, below the oculomotor and trochlear nerves. Just before entering the orbit, through the superior orbital fissure, it divides into 3 branches, lacrimal, frontal, and nasociliary. The ophthalmic nerve is joined by filaments from the cavernous plexus of the sympathetic, and communicates with the oculomotor, trochlear, and abducent nerves. It gives off a recurrent filament which passes between the layers of the tentorium. The lacrimal nerve, nervus lacrimalis, is the smallest of the 3 branches of the ophthalmic. It sometimes receives a filament from the trochlear nerve, but this is possibly derived from the branch which goes from the ophthalmic to the trochlear nerve. It passes forward in a separate tube of dura mater, and enters the orbit through the narrowest part of the superior orbital fissure. In the orbit it runs along the upper border of the rectus lateralis with the lacrimal artery, and communicates with the zygomatic branch of the maxillary nerve. It enters the lacrimal gland and gives off several filaments, which supply the gland and the conjunctiva. Finally, it pierces the orbital septum and ends in the skin of the upper eyelid, joining with filaments of the facial nerve. The lacrimal nerve is occasionally absent, and its place is then taken by the zygomatico-temporal branch of the maxillary. Sometimes the latter branch is absent, and a continuation of the lacrimal is substituted for it. The frontal nerve, nervus frontalis, is the largest branch of the ophthalmic, and may be regarded, both from its size and direction, as the continuation of the nerve. It enters the orbit through the superior orbital fissure, and runs forward between the levator palpebrae superioris and the periosteum. Midway between the apex and base of the orbit it divides into 2 branches, supratrochlear and supraorbital. The supratrochlear nerve, nervus supratrochlearis, is the smaller of the 2, passes above the pulley of the obliquus superior, and gives off a descending filament to join the infratrochlear branch of the nasociliary nerve. It then escapes from the orbit between the pulley of the obliquus superior and the supraorbital foramen, curves up onto the forehead close to the bone, ascends beneath the corrugator and frontalis, and dividing into branches which pierce these muscles, it supplies the skin of the lower part of the forehead close to the middle line, and sends filaments to the conjunctiva and skin of the upper eyelid. The supraorbital nerve, nervus supraorbitalis, passes through the supraorbital foramen, and gives off, in this situation, palpebral filaments to the upper eyelid. It then ascends upon the forehead and ends in 2 branches, a medial and a lateral, which supply the integument of the scalp, reaching nearly as far back as the lambdoidal suture. They are at first situated beneath the frontalis, the medial branch perforating the muscle, the lateral branch the gallia aponeurotica. Both branches supply small twigs to the pericranium. The nasociliary nerve, nervus nasociliaris, nasal nerve, is intermediate in size between the frontal and lacrimal, and is more deeply placed. It enters the orbit between the 2 heads of the rectus lateralis, and between the superior and inferior rami of the oculomotor nerve. It passes across the optic nerve and runs obliquely beneath the rectus superior and obliquus superior to the medial wall of the orbital cavity. Here it passes through the anterior ethmoidal foramen, and, entering the cavity of the cranium, traverses a shallow groove on the lateral margin of the front part of the cribriform plate of the ethmoid bone, and runs down, through a slit at the side of the crista galli, into the nasal cavity. It supplies internal nasal branches to the mucous membrane of the front part of the septum and lateral wall of the nasal cavity. Finally, it emerges as the external nasal branch between the lower border of the nasal bone and the lateral nasal cartilage and, passing down beneath the nasalis muscle, supplies the skin of the ala and apex of the nose. The nasociliary nerve gives off the following branches, viz., the long root of the ciliary ganglion, the long ciliary, and the ethmoidal nerves. The long root of the ciliary ganglion, radix longa ganglii ciliaris, usually arises from the nasociliary between the two heads of the rectus lateralis. It passes forward on the lateral side of the optic nerve and enters the posterior-superior angle of the ciliary ganglion. It is sometimes joined by a filament from the cavernous plexus of the sympathetic, or from the superior ramus of the trochlear nerve. The long ciliary nerves. Nervus ciliaris longi, 2 or 3 in number, are given off from the nasociliary as it crosses the optic nerve. They accompany the short ciliary nerves from the ciliary ganglion, pierce the posterior part of the sclera, and running forward between it and the choroid are distributed to the iris and cornea. The long ciliary nerves are supposed to contain sympathetic fibres from the superior cervical ganglion to the dilator pupillae muscle. The infratrochlear nerve, nervus infratrochlearis, is given off from the nasociliary just before it enters the anterior ethmoidal foramen. It runs forward along the upper border of the rectus medialis and is joined, near the pulley of the obliquus superior, by a filament from the supratrochlear nerve. It then passes to the medial angle of the eye and supplies the skin of the eyelids and side of the nose, the conjunctiva, lacrimal sac, and caruncula lacrimalis. The ethmoidal branches, nervus ethmoidalis, supply the ethmoidal cells. The posterior branch leaves the orbital cavity through the posterior ethmoidal foramen and gives some filaments to the sphenoidal sinus. The ciliary ganglion, ophthalmic or lenticular ganglion. The ciliary ganglion is a small sympathetic ganglion of a reddish-grey colour and about the size of a pin's head. It is situated at the back part of the orbit, in some loose fat between the optic nerve and the rectus lateralis muscle, lying generally on the lateral side of the ophthalmic artery. Its roots are 3 in number, and enter its posterior border. 1, the long or sensory root, is derived from the nasociliary nerve, and joins its posterior-superior angle. The second, the short or motor root, is a thick nerve, occasionally divided into 2 parts, derived from the branch of the oculomotor nerve to the obliquus inferior, and connected with the posterior-inferior angle of the ganglion. The motor root is supposed to contain sympathetic efferent fibres, preganglionic fibres, from the nucleus of the third nerve in the midbrain to the ciliary ganglion, where they form synapses with neurons whose fibres, postganglionic, pass to the ciliary muscle and to the sphincter muscle of the pupil. The third, the sympathetic root, is a slender filament from the cavernous plexus of the sympathetic. It is frequently blended with the long root. According to Tiedemann, the ciliary ganglion receives a twig of communication from the sphenopalatine ganglion. Its branches are the short ciliary nerves. These are delicate filaments, from 6 to 10 in number, which arise from the forepart of the ganglion in 2 bundles connected with its superior and inferior angles. The lower border is the larger. They run forward with the ciliary arteries in a wavy course, one set above and the other below the optic nerve, and are accompanied by the long ciliary nerves from the nasociliary. They pierce the sclera at the back part of the bulb of the eye, pass forward in delicate grooves on the inner surface of the sclera, and are distributed to the ciliaris muscle, iris, and cornea. Tiedemann has described a small branch as penetrating the optic nerve with the arteria centralis retinae. The maxillary nerve, nervus maxillaris, superior maxillary nerve, or second division of the trigeminal, is a sensory nerve. It is intermediate, both in position and size, between the ophthalmic and the mandibular. It begins at the middle of the semilunar ganglion as a flattened plexiform band, and, passing horizontally forward, it leaves the skull through the foramen rotundum, where it becomes more cylindrical in form and firmer in texture. It then crosses the pterygopalatine fossa, inclines lateralward on the back of the maxilla, and enters the orbit through the inferior orbital fissure. It traverses the infraorbital groove and canal in the floor of the orbit, and appears upon the face at the infraorbital foramen. At its termination, the nerve lies beneath the quadratus labii superioris, and divides into a leash of branches which spread out upon the side of the nose, the lower eyelid, and the upper lip, joining with filaments of the facial nerve. Branches. Its branches may be divided into 4 groups, according as they are given off in the cranium, in the pterygopalatine fossa, in the infraorbital canal, or on the face. In the cranium, middle meningeal. In the pterygopalatine fossa, zygomatic, sphenopalatine, posterior superior alveolar. In the infraorbital canal, anterior superior alveolar, middle superior alveolar. On the face, inferior palpebral, external nasal, superior labial. The middle meningeal nerve, nervus meningeus medius, meningeal or dural branch, is given off from the maxillary nerve directly after its origin from the semilunar ganglion. It accompanies the middle meningeal artery and supplies the dura mater. The zygomatic nerve, nervus zygomaticus, temporomalar nerve, orbital nerve, arises in the pterygopalatine fossa, enters the orbit by the inferior orbital fissure, and divides at the back of the cavity into 2 branches, zygomatico-temporal and zygomatico-facial. The zygomatico-temporal branch, ramus zygomatico-temporalis, temporal branch, runs along the lateral wall of the orbit in a groove in the zygomatic bone, receives a branch of communication from the lacrimal, and, passing through a foramen in the zygomatic bone, enters the temporal fossa. It ascends between the bone and the substance of the temporalis muscle, pierces the temporal fascia about 2.5 cm above the zygomatic arch, and is distributed to the skin on the side of the forehead, and communicates with the facial nerve and with the auriculate, ulotemporal branch of the mandibular nerve. As it pierces the temporal fascia, it gives off a slender twig, which runs between the 2 layers of the fascia to the lateral angle of the orbit. The zygomatico-facial branch, ramus zygomatico-facialis, malar branch, passes along the infralateral angle of the orbit, emerges upon the face through a foramen in the zygomatic bone, and, perforating the orbicularis oculi, supplies the skin on the prominence of the cheek. It joins with the facial nerve and with the inferior palpebral branches of the maxillary. The sphenopalatine branches, nervus sphenopalatini, 2 in number, descend to the sphenopalatine ganglion. The posterior superior alveolar branches, ramae alveolares superiores posteriores, posterior superior dental branches, arise from the trunk of the nerve just before it enters the infraorbital groove. They are generally 2 in number, but sometimes arise by a single trunk. They descend on the tuberosity of the maxilla, and giving off several twigs to the gums and neighbouring parts of the mucous membrane of the cheek. They then enter the posterior alveolar canals on the infratemporal surface of the maxilla, and, passing from behind forward in the substance of the bone, communicate with the middle superior alveolar nerve, and give off branches to the lining membrane of the maxillary sinus and 3 twigs to each molar tooth. The twigs enter the foramina at the apices of the roots of the teeth. The middle superior alveolar branch, ramus alveolaris superior medius, middle superior dental branch, is given off from the nerve in the posterior part of the infraorbital canal, and runs downward and forward in a canal in the lateral wall of the maxillary sinus to supply the 2 premolar teeth. It forms a superior dental plexus with the anterior and posterior superior alveolar branches. The anterior superior alveolar branch, ramus alveolaris superior anteriores, anterior superior dental branch, of considerable size, is given off from the nerve just before its exit from the infraorbital foramen. It descends in a canal in the anterior wall of the maxillary sinus, and divides into branches which supply the incisor and canine teeth. It communicates with the middle superior alveolar branch, and gives off a nasal branch, which passes through a minute canal in the lateral wall of the inferior meatus, and supplies the mucous membrane of the anterior part of the inferior meatus and the floor of the nasal cavity, communicating with the nasal branches from the sphenopalatine ganglion. The inferior palpebral branches, rami palpebrales inferiores, palpebral branches, ascend behind the orbicularis oculi. They supply the skin and conjunctiva of the lower eyelid, joining at the lateral angle of the orbit with the facial and zygomaticofascial nerves. The external nasal branches, rami nasalis externi, supply the skin of the side of the nose and of the septum mobili nisi, and join with the terminal twigs of the nasociliary nerve. The superior labial branches, rami labiali superiores, labial branches, the largest and most numerous, descend behind the quadratus labii superioris, and are distributed to the skin of the upper lip, the mucous membrane of the mouth, and labial glands. They are joined immediately beneath the orbit by filaments from the facial nerve, forming with them the infraorbital plexus. Sphenopalatine ganglion, ganglion of Meckel. The sphenopalatine ganglion, the largest of the sympathetic ganglia associated with the branches of the trigeminal nerve, is deeply placed in the pterygopalatine fossa, close to the sphenopalatine foramen. It is triangular or heart-shaped, of a reddish-grey colour, and is situated just below the maxillary nerve as it crosses the fossa. It receives a sensory, a motor, and a sympathetic root. Its sensory root is derived from two sphenopalatine branches of the maxillary nerve. Their fibres, for the most part, pass directly into the palatine nerves. A few, however, enter the ganglion constituting its sensory root. Its motor root is probably derived from the nervus intermedius through the greater superficial petrosal nerve, and is supposed to consist in part of the sympathetic efferent preganglionic fibres from the medulla. In the sphenopalatine ganglion the form synapses with neurons whose postganglionic axons, vasodilator and secretory fibres, are distributed with the deep branches of the trigeminal to the mucous membrane of the nose, soft palate, tonsils, uvula, roof of the mouth, upper lip and gums, and to the upper part of the pharynx. Its sympathetic root is derived from the carotid plexus through the deep petrosal nerve. The two nerves join to form the nerve of the pterygoid canal before their entrance into the ganglion. The greater superficial petrosal nerve, nervus petrosus superficialis major, large superficial petrosal nerve, is given off from the genicular ganglion of the facial nerve. It passes through the hiatus of the facial canal, enters the cranial cavity, and runs forward beneath the dura mater in the groove on the anterior surface of the petrous portion of the temporal bone. It then enters the cartilaginous substance which fills the foramen lacerum, and joining with the deep petrosal branch forms the nerve of the pterygoid canal. The deep petrosal nerve, nervus petrosus profundus, large deep petrosal nerve, is given off from the carotid plexus, and runs through the carotid canal lateral to the internal carotid artery, then enters the cartilaginous substance which fills the foramen lacerum, and joins with the great superficial petrosal nerve to form the nerve of the pterygoid canal. The nerve of the pterygoid canal, nervus canalis pterygoidei, vidii, Vidian nerve, formed by the junction of the two preceding nerves in the cartilaginous substance which fills the foramen lacerum, passes forward through the pterygoid canal with the corresponding artery, and is joined by a small ascending sphenoidal branch from the otic ganglion. Finally, it enters the pterygopalatine fossa, and joins the posterior angle of the sphenopalatine ganglion. End of Part 28.